Hello and welcome, students. Today we're stepping into a new practical lesson. Today, we're going to dive into an experiment that ties together our material in plant transpiration and gas exchange. We're about to head into our lab to explore a fascinating experiment that directly ties into the concepts we've been covering. But first, over to Sam, our lab technician, to outline what safety equipment we need.
Hi everyone, I'm Sam. Before we get started, make sure to put on your lab coat, safety goggles and gloves. Remember, safety is our top priority in any lab environment.
Thanks, Sam. While we're getting ready, let's quickly recap what we've learned so far. So far, we've explored the various plant organs and their specific functions, particularly focusing on the leaves, roots, stems, and flowers. We've studied how these organs work together to ensure water and nutrients are transported throughout the plant. You've learned about the xylem and phloem, the crucial vascular tissues responsible for this transportation. We also discussed the concepts of transpiration and how water evaporates from the leaves, creating a negative pressure that helps draw water up through the plant, and the factors that affect the rate of transpiration. Today's practical will put this knowledge into action as we measure the rate of transpiration by observing the uptake of water in plants. This experiment will offer you a hands-on experience to see how water moves through the xylem during transpiration, reinforcing the principles we've discussed. Understanding the rate of transpiration is essential as it influences how efficiently a plant can transport essential nutrients and maintain its water balance in response to its environment. Before outlining the practical, let's take a moment to consider its importance. Understanding the rate of transpiration in plants is crucial for both ecological studies and agriculture. By measuring the rate of transpiration, you can infer how well a plant is adapting to its environment, including how it responds to changes in humidity, temperature, and availability of water. In agriculture, knowing the rate of transpiration helps farmers optimize irrigation practices, ensuring that crops receive the right amount of water, which can significantly impact crop yield and the efficient use of water resources. Secondly, this practical offers you the opportunity to refine essential scientific skills such as precise measurement, data collection, and analysis. By using a potometer to measure water uptake, which is an indirect way of assessing the rate of transpiration, you learn to handle scientific equipment accurately and interpret data effectively. Finally, measuring the rate of transpiration helps deepen your understanding of plant physiology, specifically water transport mechanisms. The experiment demonstrates how xylem vessels in plants facilitate the movement of water from the roots to the leaves through the transpiration stream. Now we understand its importance. Let's dive in and investigate how transpiration rates can be measured through water uptake. Before we get started, let's quickly review the equipment we need. Sam, take us through the list.
For this experiment, you will need a plant, some sharp scissors, a potometer, a lamp, a fan, a temperature dial to change the room temperature, a plastic bag, and finally, a lab book.
Great. Let's get started. For this practical, it's essential to maintain a precise setup to ensure reliable results. So let's go step by step. First, we need to carefully cut a shoot from our chosen plant. This must be done underwater to prevent air from entering the xylem, which could disrupt the water flow. Make sure to use sharp scissors and handle the plant delicately to avoid damage. Next, we are going to set up our potometer apparatus. This involves placing the plant shoot into the potometer tube and ensuring that all joints and connections are sealed tightly with Vaseline to make the system airtight. It's crucial that no air leaks into the system, as this would compromise our readings. Once the setup is complete, we need to introduce an air bubble into the capillary tube of the potometer. To do this, gently remove the capillary tube from the beaker of water, allowing a single air bubble to form, and then place the tube back into the water. Be careful with this step to ensure the bubble is accurately placed. Now, record the starting position of the air bubble using a ruler. This will be our baseline to measure the movement of the bubble over time, which indicates the water uptake by the plant. Allow the apparatus to sit undisturbed for a set period of time. This could be something like 10 or 15 minutes. During this time, the plant will continue to transpire, causing the bubble to move. After the observation period, record the final position of the air bubble. The distance the bubble has travelled corresponds to the amount of water taken up by the plant. Next, we must repeat the experiment to ensure that your results are consistent and reliable. Multiple trials help to eliminate anomalies and provide more accurate data. During the repetitions of the experiment, you can modify one variable while keeping everything else constant. This will allow us to observe which factors shape the rate of transpiration. For instance, change the light intensity by adjusting a lamp's distance from the plant, alter wind speed by using a fan at a high speed, modify humidity by wrapping the plant in a damp plastic bag, or adjust the room temperature. There are several precautions you must take to ensure this is a fair test, and that the only thing changing is the selected environmental factor. For instance, you must ensure that all connections in the potometer are airtight to avoid leaks. You must cut the plant stem underwater to prevent air from entering the xylem, and you must use fresh plant cuttings to maintain high transpiration rates. Once you have completed the repetitions with different independent variables, we can analyze our results. The central biological process we are investigating is transpiration, which is the loss of water vapor from the aerial parts of plants, primarily through small openings in the leaves called stomata. Transpiration plays a crucial role in the movement of water and nutrients from the roots to the leaves and helps in cooling the plant. Now, let's go over some of the key steps of the practical and their importance. Firstly, we cut the shoot underwater to prevent air from entering the xylem vessels, which could disrupt water uptake by creating airlocks in the transpiration stream that obstructs the flow of water. We also set up the apparatus using a potometer. This is because a potometer measures the amount of water a plant takes up, which we can use as an estimate for the rate of transpiration. It is crucial for this apparatus to be set up as shown in the diagram to obtain reliable measurements. We use Vaseline to ill gaps in the potometer. Sealing any gaps with Vaseline ensures that the apparatus is airtight, preventing air from entering the system, which could alter the results and affect the accuracy of our measurements. Furthermore, introducing a single air bubble in the capillary tube acts as a marker to measure the distance it travels over time, reflecting the volume of water taken up by the plant, and hence, transpiration rate. We were also careful to record the air bubble's starting and ending position. Noting the initial and final position of the air bubble is essential for measuring the distance it travels, which indicates the volume of water uptake over a specified period. Then, we left the apparatus for a set period of time. This allowed the experiment to run for a fixed duration, which ensures consistency in time intervals across different trials, making the measurements comparable. Next, we repeated the experiment, which ensures the reliability and reproducibility of the results. Multiple trials help in identifying anomalies and reduce the error of the measurements. After this, we began to change one factor at a time. Only changing one factor, such as light intensity, temperature, humidity, and wind speed, ensures that any changes to transpiration rate that occur are only because of this factor. Make sure all other factors are kept the same to prevent them from confounding the results. Let's now analyze the results of our experiment. Firstly, we need to measure the distance traveled by the air bubble in the potometer. To do this, subtract the starting position from the end position. Next, we can calculate the average rate of transpiration. Average the distances traveled in multiple trials to get a more accurate measure. Then use the formula to determine the rate of transpiration, which is rate of transpiration equals distance traveled by air bubble divided by time taken. For example, if the air bubble traveled 5 centimeters in 30 minutes, the rate would be 5 centimeters divided by 30 minutes equals 0.167 centimeters per minute. Once we have an average rate of transpiration that occurs without any changed factors, we can calculate the rate of transpiration that occurs when we change one factor, such as light intensity, wind speed, humidity, or temperature, while keeping all other factors constant. This gives us the opportunity to compare rates of transpiration under different conditions. Finally, we can plot a graph with the environmental condition, such as light intensity, on the x-axis and the rate of transpiration on the y-axis. This visual representation helps identify how each factor affects transpiration rate. In this case, we can see that increasing light intensity increases the rate of transpiration at first, before plateauing at around 0.18 centimeters per minute. Using the graphs, we can correlate these trends with the theory behind factors affecting transpiration. Be sure to identify any data points that do not fit the expected pattern. Consider possible causes for these anomalies, such as experimental errors or natural variations in the plant material used. Great work. That's the end of our practical. In today's lesson, we conducted a practical experiment to measure the rate of transpiration by the uptake of water in plants. Our key takeaways for future practicals are to control and accurately document environmental variables to ensure reliable and comparable results, and appreciate the relationship between environmental conditions and their impact on biological processes like transpiration. This practical reinforces our understanding of how plants manage water loss and the importance of environmental factors in influencing transpiration rates. Additionally, it highlights the need for precision in setting up experiments to achieve accurate and meaningful results.
Watch the rest of the GCSE biology course at Sophos Education.